Hello everyone, a very good evening to all of you. So how are you? I hope you all are doing great. So I'm again here with you to tell you a very important and basic concept of embryonic origin. See, this session is going to be very important for the class 11 students. Those students which have just passed the class 10th and they have come into the class 11th, for them they will be studying first animal kingdom and they will be also studying animal tissues. In their curriculum, these topics are there. So they find difficulty in understanding when we are telling that this animal tissue is derived from this germ layer. That means we are talking about the origin of every tissue, every organ of your body. So from where in your body there are so many tissues, there are so many organs, organ systems are getting formed by the different organs. So how all these organs and how all these tissues are getting formed? So this is a very basic concept which we will be studying today. So let's get started. So now see, I will start talking about the egg and sperm, everyone. I will talk about the egg. You understand egg or ovum, right? Egg or ovum is the germ cell produced by the female, right? By the female reproductive system, by the ovaries of the female, the egg is getting produced. So egg or ovum is a haploid cell, everyone, haploid cell. Okay, that means if we talk about humans, if it is haploid, that means it has 23 chromosomes in it, right? Then if we talk about sperm, so sperm is that germ cell which is produced by the male reproductive system. In the testis of the male, sperms are getting produced, right? So sperm is also a haploid cell. So when the egg and sperm, when they both will fuse, they will form a diploid structure, that is a very important structure known as zygote. Alright, so zygote is a diploid structure. 2N stands for the diploid. So that means how many chromosomes will be there in the zygote? 46 chromosomes will be there in the zygote. 23 came from the ovum and 23 came from the sperm, right? So now what is going to happen after this? See, this zygote is a single cell. It is a unicellular structure. It is just a single cell structure which is having 46 chromosomes. So unicellular structure. It is a unicellular structure. Okay, and the process of formation of zygote is known as it is known as fertilization. So here fertilization is happening. Fertilization. So due to the process of fertilization, the zygote has formed, right? Now what will happen after the zygote? This unicellular structure starts dividing. It starts division, and this division is known as cleavage. Very important. This is known as cleavage. This is known as cleavage. So what is this cleavage? Cleavage is a division which is mitotic type division. Okay, this division is mitotic division. This is a mitotic division. Do you know what is mitosis and what is meiosis? Mitosis is that division in which the chromosome number will be equal in the cell which is going to form after the division, right? But in case of meiosis, the number of chromosomes are going to be divided by 2. That means they are going to be half. If meiosis is happening in a diploid cell, then we will get haploid cells. But if mitosis is happening in the diploid cell, then we will get diploid cells only, right? So now, due to this cleavage, we will get from this unicellular structure, we will get a 2-cell structure. This is the nucleus of one cell, this is the nucleus of another cell. So can you see here two cell structure? This is a two cell structure. This is a two celled structure. So after the zygote, all the structures which are getting formed, they are known as embryo. They all are the embryonic stages. Alright. So now again the division will occur and this 2 cell stage will be converted into the 4 cell stage. Okay. Will be converted into the 4 cell stage. Then the 4 cell structure is again going to show the cleavage and then it will be converted into the 8 cell structure. 8 cell structure. Can you see this 8 cell structure? Almost the size of all these embryos is going to be the same. Whether there are 2 cells, whether there are 4 cells, whether there are 8 cells, okay. Then after 8 cell structure, this is a 8 cell structure. Again, the structure which is going to be formed is having 16 cells beta, 16 cells. So I am not going to count the cells, just I am going to make the cells, okay. I am not counting the cells. So just consider it a 16 celled structure. If you will see a 16 celled structure, this structure we have given a name to this structure, okay. We have given a name to this structure. 18 to 16 celled structure is known as morula, everyone. It is known as morula. So it is very important to know what is the next structure which is getting formed after the zygote. Okay, after the zygote, morula is going to be formed after so many divisions, after so many cleavage. All these divisions are known as cleavage, right? Then after the morula, the structure which is going to be formed is known as blastula, 32 celled structure, 32 celled structure. If you will see a blastula, then you will see a cavity in the blastula, okay. You will see a cavity in the blastula like this. So here this is the cavity in the blastula and all these are the cells in the blastula. So how many cells are there in the blastula? 32 celled stage. This is this 32 celled stage. This is the 32 celled stage and it has a name that is blastula, everyone. It has a name that is known as blastula. Alright. Blastula. So after the blastula has formed, now what will happen? This is the 32 cell stage blastula. A characteristic feature of this structure is a cavity that is known as blastocele. Here there is a cavity, you can see this cavity is known as blastocele. So everything I have one image in the next slide. I will be showing you all these structures step by step. Just I am making them so that you will understand them better. Okay, so what I have told till now, I have told that after the fusion of egg and sperm, a very important structure is going to be formed which is a unicellular diploid structure, that is zygote. Then zygote will start the mitotic division, that is known as cleavage. It is not exactly same as mitosis, so that is why we say particularly we can say it is a mitotic type division. It is just like mitosis only and it is specifically occurring in the zygote and then it will occur in the further embryos as well. So zygote is a single cell structure, then by cleavage it will be converting into a 2 cell structure, then further the cleavage will happen in that embryo, then 4 cells, 8 cells, 16 cells, like that you will see that the number of cells are increasing and all these cells can also be called as blastomeres, everyone. All these cells, they have another name, that is blastomere. So the morula is having how many blastomeres? 8 to 16 blastomeres are there, okay. Blastula is having how many blastomeres? Blastula is having 32 blastomeres, right. So after zygote we have seen the another name morula. After morula we have seen blastula. Now a very important thing will take place, and I have told you the characteristic feature of morula and blastula. Morula, you are not seeing any cavity in the morula. Morula is a solid ball of cells, but blastula is a hollow ball of cells. It is a hollow ball, that means it has so many cells and it is hollow from inside. So that space, that cavity is known as blastocele. Alright. Then after this, what will happen after this? After this blastula, a very important process will occur, that is known as gastrulation. So what is this gastrulation, everyone? Gastrulation is the process of formation of embryonic germ layers. Gastrulation is the process of formation of embryonic germ layers. Very important. What is the meaning of germ layers? Germ layers means they are going to give rise to everything in your body. Okay, everything. So all the tissues and organs are formed by these germ layers. They are germinating, they are giving rise to all the tissues and organs of the body, right? So process of formation of embryonic germ layers by very important, by morphogenetic movements. What are these movements? We have seen so many cells in the blastula, everyone? Yes or no? Please tell me in the comments if you are able to understand this process of embryonic division, embryonic origin. Alright. So what is gastrulation? Process of formation of embryonic germ layers by morphogenetic movements. What are these movements? We will not go into the details of these movements. There are so many movements. So basically morpho means form. Okay, the form is changing, basically the embryo is changing the form. The cells are moving in such a way that they are giving rise to the three germ layers. The three germ layers, if I talk about triploblastic animals, three germ layers are getting formed. So what are these three germ layers, everyone? These are ectoderm, mesoderm and endoderm. So when these three germ layers are formed in the blastula, then we will say the next structure has developed, and that is known as gastrula. So by the process of gastrulation, the next structure which is going to be formed, that is known as gastrula. So now you have to remember the sequence. After the zygote, what has formed? Morula. After morula, what has formed? Blastula. And after blastula, which structure has formed? Gastrula. This sequence you have to remember, and then you can tell in the gastrula we have seen three germ layers. We have seen the formation of three germ layers. They are known as ectoderm, mesoderm and endoderm. So whenever we say that our heart is mesodermal in origin, so that means our heart is formed by which germ layer, everyone? It is formed by mesoderm layer. So now when you will be studying animal tissues, or you will be studying animal kingdom, you will not be getting confused if we will say coelom. Coelom is formed by mesoderm layer. So you will understand that mesoderm is a germ layer. It has formed during the embryonic life. It has formed after the 32 celled stage, that is blastula. So after the formation of the blastula, these germ layers have developed. Alright. So now if I will tell you, animal tissues are of four types. Okay. I am just briefing it that animal tissues are of four types. One is epithelial tissue. Everyone. One is connective tissue, which is the most abundant tissue of your body. Then there is muscular tissue. Muscular tissue is forming the muscles, and then there is nervous tissue. So let me tell you now, if I will say epithelial tissue can be formed by all the three germ layers. The origin of epithelial tissue can be ectodermal, can be mesodermal and can be endodermal. So if I will say this, then you will understand. Then if I will say connective tissue and muscular tissue, they are getting formed by the mesoderm. So the origin, now you are understanding the meaning of origin. If I will say origin of connective tissue is mesodermal, so you are understanding that where is this mesoderm, from where it has formed, after what structure it has formed. Is it present in us now or not? No, it has given rise to all the structures in your body, all the tissues and organs in your body. Okay, so now we don't have germ layers. Now the germ layers are not present in us. When we were an embryo, at that time the germ layers have formed, and then all these germ layers, all the 3 germ layers, they have given rise to all the tissues and organs. So now we can simply say about the nervous tissue also, it is derived from the ectoderm layer. Similarly, if we talk about animal kingdom, in animal kingdom you will be studying so many structures, like you will be studying coelom, you will be studying notochord. Okay, both of these structures are derived from the mesoderm layer, So I have included a table in it, okay, and you will get all these notes on the telegram. So please join the telegram. If you will see the description of this video, so then you will find the telegram channel. So please join it. You will find a link there. Please join it and you will get all these notes there. Okay. So I have included also in your body, if you want to know in your body which structures are formed by which germ layers. So we will be providing that. Okay. Now just revising it quickly, then we will be done. So just revising it quickly, if you will see the human embryonic development, can you see that sperms are coming near to the ovum? This is ovum, these are sperms, and obviously only one sperm is going to fuse with the ovum, alright. So then the egg will become fertilized egg, okay. Then this fertilized egg is basically zygote. Fertilized egg is basically zygote has formed. So then you can see here 2-celled stage, then you can see 4-celled stage. So at every stage what is happening? Division is happening, and that division is known as cleavage. Here the first cleavage, the second cleavage, like that. You will understand all these cleavages in detail. You will study in the human reproduction chapter in class 12th. But before that, why I am explaining all this for the 11th class students, that if they will understand this topic, then it will be very easy for them to understand animal tissues and animal kingdom and other things where we are talking about the origin. Alright. So after this cleavage you can see 4-cell structure, then again the cleavage will happen, 8-cell structure. So here can you see the solid ball of cells? This is the solid ball of cells, This is known as morula, everyone, morula. Then after morula you can see blastula here, 32-celled structure, 32 cells are there. This is the 32-celled stage. So after 16-cell stage, 32-cell stage will come. In morula there will be 16 cells. Then again the early gastrula you can see. So can you see some movements of the cells? If you are able to see some movements, these movements are known as morphogenetic movements. Alright, morphogenetic movements. So due to these movements, there will be formation of, you can see ectoderm here, you can see endoderm here, you can see a cavity, and the opening of that cavity is known as blastopore. You can see the primitive gut here, archenteron, and this whole structure is known as gastrula. In between ectoderm and endoderm, mesoderm is going to be formed. Alright. So all these 3 germ layers are going to be formed after the blastula. When all these germ layers will form, that structure we will call it gastrula. So I hope you have understood this. Now you can just revise quickly that this ectoderm, mesoderm and endoderm is forming what all organs and tissues in your body. If we talk about your skin, your epidermis, it is derived by the ectoderm. Your sweat glands which are present in your skin, These are derived from the ectoderm. Okay. Then the epithelial lining of your gut is derived from the ectoderm. The cornea and the lens of your eye, the nervous system, I have just now told you that the nervous tissue is derived from the ectoderm. So, nervous system, receptors, epidermis, there is a adrenal gland in which there are two parts, one is adrenal medulla, one is adrenal cortex. So, adrenal medulla is derived from the ectoderm. Tooth enamel is derived from the ectoderm. Epithelial of your pineal gland, of your pituitary gland is derived from your ectoderm. Similarly, if you will see, just now I have told you notochord, yes, just now I have told you muscle, just now I have told you connective tissue. So, skeletal system is connective tissue. Then stomach and intestine, the muscles of stomach and intestine, excretory system, circulatory system, reproductive system, except the germ cells. Dermis of skin, dermis is the layer, the uppermost layer will be epidermis, then below that there will be dermis. So, epidermis is derived from the ectoderm, but dermis is derived from the mesoderm layer. Then lining of body cavity, the coelom is lined by the mesoderm. Do you know the coelom? Then adrenal cortex, just now I have told you adrenal medulla has, the adrenal gland has two parts, adrenal medulla and adrenal cortex. So, just remember adrenal medulla is ectodermal in origin and adrenal cortex is mesodermal. Similarly, if we talk about liver, pancreas, thymus, thyroid gland, parathyroid gland, your lining of urethra, urinary bladder, reproductive system, epithelial lining of your respiratory system, epithelial lining of your digestive tract, all these are derived from the endoderm. Epithelial lining of your blood vessels, that is known as endothelium. So, that epithelium which is derived from the endoderm. So, this is all about the origin, embryonic origin, where we have talked about all the three germ layers, ectoderm, mesoderm and endoderm. So, I hope you have understood this part. If you have any doubt, any difficulties in understanding this, you can comment. You can please just tell me in the comments. I will be taking your doubts there, and you can also suggest the topics in which you are facing problem. If you are a class 11 student, if you are a class 12 student, if you are facing any difficulty in any of the topic and you want me to explain it in a better way, so then you can tell me in the comments. Okay, thank you for watching this video. Thank you very much. If you like the video, please hit the like button, and for more updates you can subscribe to our channel. Thank you very much.